and welcome to the Fertility Conversations podcast. The goal of this podcast is to create more awareness about infertility and to provide support to people trying to conceive. Thank you for listening today, and we hope you will be encouraged. And now, here is your host, Ola.
Welcome to another episode of Fertility Conversations. Today, we're joined by a lovely guest, Jessica, who's here to give us some insights into the embryology part of IVF. Jessica loves helping people to build their families, and she started her platform on social media, Explaining IVF, to be able to shed more light and provide more information about IVF and infertility because it's such a complex process. Her details will also be in the show notes. So welcome, Jessica, and thank you so much for joining us today.
Thank you for having me.
Yay. So we usually start off by saying, tell us a little bit about yourself.
Okay. So my name's Jessica. I live in the United States. I've been an embryologist since 2018 and really love my job. Absolutely love helping people build their families, working in the lab. And then I started my page Explaining IVF last year, actually, to kind of help bridge some of the knowledge gaps that come with IVF.
Wonderful. Thank you. I'm so glad to have you here because, again, we always have so many questions. So to be able to have you here to answer some of the many questions we have is such a blessing. So thank you for being here. One of the many questions we have is, what exactly is the IVF timeline?
That's a good question. So the IVF timeline can sort of differ from person to person, but the time that the embryos are actually in the lab is pretty much the same everywhere. So the timeline can start at your initial consult with your IVF doctor. And then from there, it might take a couple of months before you actually begin the IVF process. And the only reason is because you might need some diagnostic tests to be done beforehand. You might require some time off for personal reasons. So just because you go to an IVF clinic and have that initial consult doesn't necessarily mean that the process will start right away, but it can. It can normally start within a month or so, just depending. But then from there, the process really takes about 2 to 3 weeks. So you would start your menstrual cycle and you'll start taking medications. In some cases, you might actually take medications beforehand, but in a lot of cases, you'll start taking medications when you start your menstrual cycle that the IVF cycle will be a part of. And those medications basically stimulate the ovaries so that multiple eggs will mature instead of just one, which is what would normally happen. And then you take those for a few days, and then you might take medications that, help control the timing of ovulation. And that's important because you want to make sure ovulation is occurring soon after your egg retrieval procedure, not before, otherwise the eggs will be lost. So that process can take anywhere from about maybe 8 to 14 days, give or take, maybe a little bit more for some people, depending on how long it takes those eggs to mature, the follicles that they're in to grow. So that process, like I said, can take anywhere from about like 8 to 14 days. And then from there, you'll have your egg retrieval scheduled. And that's where everything sort of becomes more uniform in the timeline. That's normally like a 6 to 7 day process. So you would do your egg retrieval on one day. If you're freezing your eggs, they would be frozen that day as well. Otherwise, if you're doing any insemination for embryo culture, it would also be done that same day. The next day, the embryos would be checked to make sure they fertilize properly, and then they're given a couple of days to grow. So some clinics will look at them over the course of the next couple of days, but most clinics look at them 5 days after the insemination occurred. That's called day 5 in the IVF cycle. That's when the embryos can start to be frozen. If you're doing a fresh transfer, that can occur on that day. If you're doing genetic testing, that can occur on that day. But any embryos that maybe aren't ready that day that are still growing, they'll get another day or 2 to grow, depending on how many days your clinic will culture embryos for. So that's where it varies a little bit in the lab, but for the most part, it's 6 to 7 days.
Wow. Thank you. That's so detailed. So much actually goes on that we don't realize, right? People don't realize that it can even differ because we always assume that it's the same for everyone. So it's good to know that. Thank you. And even with the IVF, sometimes people wonder, what is ICSI and how is it different from the standard IVF? And should we be doing ICSI all the time? Is it for everyone, or can you shed some light on that?
Absolutely. And there are actually newer studies that are coming out. So I'll talk a little bit about those too. But for the most part, there are two types of insemination that can occur. One is called ICSI and the other is called conventional or standard IVF, or conventional insemination. It has a couple of different names. So ICSI refers to taking one sperm and injecting it into one mature egg. And so that's a way to sort of first force fertilization to occur. And then after all of the eggs have been inseminated with these sperm, they go into the incubator overnight and they're checked the following day for fertilization. With the other form, the conventional or standard insemination, or IVF as it's called, the sperm and eggs are all put into a couple of drops together, and they're allowed to sort of interact and hopefully fertilize overnight. So the problem is with that type of insemination, egg grading is not taken into account. So just briefly to touch on that, with ICSI, you actually clean the eggs before the insemination and you grade them to see if they're mature. So only eggs that are mature are the ones that are usually ICSI'd. Whereas with the other form of insemination, they're not graded beforehand. So all of the eggs go in with the sperm. But if they're not mature, they won't fertilize properly. So you can expect a lower fertilization rate with the standard or conventional method, but it's probably because some of the eggs that are going in there are not mature, but we don't know that going into it. So both methods are okay if there are no male factor issues at play. If there are male factor issues such as a low sperm count, low motility, low morphology, maybe a high, DNA fragmentation, or if there's maybe unexplained infertility in the past or poor fertilization in the past, then you might want to turn to ICSI. And the reason for that is because you only need enough sperm for every mature egg with ICSI, whereas for the other form, you need a lot more sperm for them to be able to get to the egg and perform the fertilization.
Wow, thank you. It's good to know that, because sometimes we all just want to do ICSI because we hear about it and you're like, oh, okay, maybe I should be doing ICSI, but you might not be required based on your individual situation.
Right. And that's where those new studies are coming in. They're saying, when there is no male factor infertility at play, or no, previous poor fertilization or other sort of underlying conditions, that standard insemination it should yield the same results as the ICSI. But again, there are situations where ICSI is absolutely needed. Otherwise, your fertilization rate will be very poor with the conventional method. And I think that's where ICSI became so popular. A lot of clinics just do pretty much 100% ICSI. And really, it's because it's a way to, again, force fertilization to occur. So that at least insemination is occurring, and then you're hoping that fertilization occurs as well. But it's a way to know that insemination is at least occurring. Whereas with the other method, you're hoping that the eggs are mature, you're hoping that the sperm get to the egg, you're hoping they inseminate properly and fertilize properly.
Thank you. Thank you for sharing that. We know so much more now and have a better understanding. And with the egg retrieval, sometimes, I've had cycles before where there was no eggs retrieved. Until you wonder, because there were follicles when you were doing the scans, you could see follicles going into the egg retrieval. Then you come out and there is no eggs. So you wonder, how could that have been when there were, seemingly big follicles that eggs should be in? So can you explain why that can happen? Or?
Yeah, absolutely. I know it's really frustrating. I know it's absolutely frustrating for the patient, but it's frustrating for us too, because you spend all this time doing your monitoring, taking your medications, you have these follicles, you go in with expectations, and then, nothing. So yes, very frustrating. There are a couple of reasons why that can occur. So one reason might be that ovulation either occurred early or was occurring maybe right around the time of the process. So what that means is that follicle still might be there, but the egg was already ovulated. And sometimes it's because maybe their medication wasn't taken at the right time. It might be that maybe the procedure was very, very late, that maybe they were backed up a couple of hours, which isn't common, but it can happen. So those are a couple of reasons why ovulation might occur. And sometimes even when you take the trigger, some people just still ovulate early, even though it's uncommon. I have seen that happen. And unfortunately, once ovulation occurs, the eggs can't be retrieved because they're now floating basically into the fallopian tube, and they just can't be retrieved via the egg retrieval. Another reason might just be a poor response the medication or an abnormal response to the medication. And what happens there is the follicles grow, but the eggs inside of them either are not mature, or they degenerate, or there's no egg there at all. And when we see that a lot, it's with a condition known as diminished ovarian reserve, which means that the women don't have a lot of eggs remaining in their ovaries. We see that a lot with women who are maybe in their 40s. And what happens is the follicles still might be there, but the eggs inside have either degenerated or they're not there, or again, they just did not mature properly. So maybe an egg is retrieved, but it's still not mature, meaning it's unusable. And then the other thing could be that the egg still adhered to the follicle wall. So when the egg is inside of the follicle, it adheres to the wall of the follicle. And as it becomes more mature, it's easier for it to aspirate or suck up the fluid with the egg inside of it. But what can happen sometimes is even when you aspirate that fluid, the egg still sticks to the inside of the follicle wall. And unfortunately, the eggs are too small to see on the ultrasound. So once you take the fluid, you really don't know if the egg's in there or not. And you really can't go back in and, try to get the egg out once the fluid is aspirated.
Wow. Sounds really complicated. So much goes into it.
Yeah.
So
yes,
it's
frustrating.
it's actually like a miracle when things go right, because there can be so many things that could go wrong.
It's at every step of the process. I completely agree.
Wow. And so if you're able to get the eggs and then the sperm is fine and you fertilize, how do you then grade the embryos?
So some clinics, again, they do look at the embryos on normally day 3 is a common time to look at the embryos. Some clinics do it. The clinics I worked at do not. But for the clinics that do, they'll take the embryos out on day 3 just briefly for a couple of moments, and they'll look to make sure that the cells are dividing. So at that point, you want to see at least 6 cells, if not more, in the embryo. You want to make sure that there's not a lot of fragmentation. And that means that the cells are trying to divide, but instead of dividing into 2 even cells, they're breaking apart into smaller pieces. Some fragmentation is perfectly fine, but if there's, if the fragmentation is taking up 80% of that embryo, it's usually not a good sign. It means that the embryo will probably not grow from there. So day 3 checks, they're good, but they don't really tell you about what will happen on day 5, meaning that you might have an embryo that's a little slower growing on day 3, and it might turn into a beautiful embryo on day 5, or vice versa. You might have this beautiful day 3 embryo that stops growing at the 8-cell stage. So it's good, but it's not definitive, is what I should say. It's more of an indicator of what they estimate will happen. But the more common embryo grading starts to kick in on day 5 or 6 or 7. And that's when we look at those embryos and we can start that whole 5AA, 5AB process. So what we see there are 3 criteria. The first criteria is that you want to make sure that the embryo is at a certain stage of development. So we look for normally around the expanded blastocyst stage. That's the number 4 if your clinic uses a numbering system. And what that means is that the embryo has divided into about 150 cells, give or take a few. And the shell around the embryo is starting to thin out as that embryo grows inside of it. We also see a giant cavity called a blastocele. It's like a fluid-filled cavity inside the embryo. And then we also have 2 distinct cell lines at that point. We have a clump of cells that sort of sits off to the side. When we look at the pictures of the embryos, we sort of see like a circle or clump of cells that sits off to one side. That's called the inner cell mass. That's the part of the embryo that ultimately will develop into the baby. And then you see cells that line the shell of the embryo, and those are called the trifectoderm cells, and those ultimately become the placenta or anything that is not the baby. And so we use a grading system. So the first one is the number. So if you have that expanded blastocyst, it would be a number 4. And then after that shell starts to thin out, a hole will actually start to form in it, and the embryo will actually start to hatch out of that shell. So almost if you think of kind of like a chicken egg, it's similar where when they're ready to hatch, they'll poke a little hole in and they'll start to hatch out of it. It's very similar. So that would be a hatching blastocyst. That would be given a number 5. And then eventually that embryo will completely hatch out of that shell. And that's when you see pictures of embryos with no shell around them, they're fully hatched. And that's a number 6 on the normal grading system, numbering system, which is called the Gardner scale. So that's the first thing that we look at. The second is that clump of cells called the inner cell mass, that part that ultimately becomes the baby. And we grade those on an ABC scale. Some clinics also use a D scale. but ABC is the most common. So A is the best grade that you can give it. That means it's really compact. It looks exactly like it's supposed to. It's like a textbook perfect inner cell mass. A B is a fair grade, which is the most common. Just because it's a B grade does not make it bad at all. B grades are very, very common. But C grades are what we call poor quality. And that means that what we're looking at is below average. Maybe the inner cell mass is there, but it's really small. Maybe the cells look a little dark or grainy. So it's what we call below average. And the same grading system is used for the trophectoderm. So those cells that line the shell of the embryo that become the placenta, we also give them an A, B, C, sometimes D grade. A would be the best, meaning it's very uniform. The cells all look very similar. There's no gaps. So textbook perfect trophectoderm. B would be fair, average, which is same criteria for the inner cell mass, and C is below average. So maybe there are no cells, big gaps, not a lot of cells, maybe they're dark and grainy. So if you have a 5AA embryo, you have a hatching embryo that has a beautiful textbook perfect inner cell mass and trophectoderm.
Wow.
Yes.
Yeah. So basically, I was saying that why is that sometimes these perfect embryos don't implant, and the ones that do not appear so perfect based on the grading system then implant. So is it because the grading doesn't really matter, or are there other factors at play as well?
So the grading is important. It can tell you, if no other variables are considered, the embryo grading can tell you which embryo has the highest chance of success. So a better looking embryo will have a higher chance of success if you don't consider anything else. But grading is only one factor when it comes to an embryo's overall success rate. That could mean that the embryo might be genetically abnormal. So even if it's a beautiful 5AA embryo, if it doesn't have the right amount of DNA in it, it will either not implant, it has a very high chance of miscarrying, or it could lead to pregnancy or birth complications or genetic defects. So that's one thing to consider, that even if your embryo looks beautiful, it doesn't necessarily mean that it's genetically normal, and vice versa. An embryo that might look below average could be genetically normal and result in a healthy live birth. Another factor, of course, when it comes to the success rates has nothing to do with the embryo. It actually has to do with the uterine environment. And that could mean that you might have the perfect embryo, but if it doesn't have the perfect uterine environment to implant in, then either implantation will not occur, or miscarriage could occur, or pregnancy complications down the road. So it's only one factor when it comes to an embryo's overall success. It is important, but it's not definitive, or can't guarantee really much of anything.
Okay, that's good to know, because I think that's something that comes up a lot in the community, and people trying to conceive, just wondering what's going on, how come this didn't implant when it seems so perfect. So it's good to know there's other issues at play. But what about mosaic embryos? What are those? And because again, people talk about that, what's a mosaic embryo and what exactly does that mean for me?
And that's a hot topic right now too, because newer studies are coming out about mosaics all the time, because they're relatively, they're relatively new. Mosaicism has only been reported on genetic testing since I think 2015. So it's a very new concept.
Yeah.
And I think a lot of the IVF community just doesn't quite know what to do with mosaic embryos right now. But luckily, a lot of the newer studies have found that at least these mosaics, which I'll talk about in a second, but at least these mosaics that kind of border on being normal actually might be okay for transfer. But a mosaic embryo is an embryo that has two different cell lines. And that means that some of the cells in the embryo have a certain genetic makeup or a certain number of chromosomes, whereas another portion of the embryo has a different genetic makeup or different chromosomes. And when that happens, it could be one of two things. It could be that the embryo, when it started as one cell, it might have had the right number of chromosomes, but whenever those cells started to divide, there was some kind of error in cell division where the DNA was not evenly dispersed between the two cells. So now you have one cell with a wrong amount of DNA, maybe it has an extra chromosome, and then and then you have an embryo over here that has a different amount of DNA or a different number of chromosomes. Those cells then go on to replicate their DNA when they have cell division. And what you end up with is this embryo with 150 cells or so, but some of a portion of the cells have this number of chromosomes, where another portion of the embryo has this number of chromosomes. And it all results from that early division where something happened that caused that to happen. That cause it's called non-disjunction, where the DNA does not evenly disperse during cell division. So at some point when that non-disjunction occurred, it caused those two different cell lines. So that's called one way that mosaicism can happen. Another way might be if the original embryo had the wrong amount of DNA. And what it does is it tries to correct that error somewhere through cell division. So somehow it's called self-correction, and the cells actually try to fix the error. But still, you're going to have cells that have the wrong DNA because that's what it started with. So that one cell divided into two, which then divides into four, but somewhere along the line, the cells tried to start correcting it. You still end up with two different portions of the embryo, one with the right amount and one with the wrong amount of DNA. So those are two ways that you can see mosaic embryos. It normally means that more than 20% of the cells in the embryo have the wrong amount of DNA. And so when we look at a genetic testing report, we have a couple of options that we see. We see euploid or normal, we see mosaic, and then we see aneuploid or abnormal. Aneuploid means that there's a lot of abnormal cells in the embryo. Normally it's about 80% or more of the cells, at least in the sample that we're taking for the biopsy, which should represent the overall genetic makeup of the entire embryo. So if most of those cells are genetically abnormal, it's considered aneuploid. And then if like 20% or less are abnormal, it's considered euploid. But then you have this gray area in the middle, which is where mosaicism stemmed from. Those embryos do tend to have lower success rates, meaning they're less likely to implant, they're more likely to miscarry. But the newer studies are finding that at least those that sort of border the normal level, so maybe they have 35% abnormal cells, maybe those ones actually have the ability to self-correct. Maybe they still might have a chance of resulting in a healthy live birth. And the studies are really interesting that are coming out, where a lot of these mosaic, low-level mosaic embryos that are being transferred are perfectly fine. They're healthy at birth, and their genetic makeup at birth is normal. So it's interesting. We're at a very important time right now with mosaic embryos where maybe they are more hopeful than we think. Now, when you get into the ones that kind of border the abnormal side, those ones still don't have good results. So they still tend to have very low success rates, and those are still a little kind of iffy in the IVF world.
Yeah. Thank you for sharing that. I think it's so important. It's good to see that there's more research going into that, because again, quite a few mosaics end up from the genetic report, and people always wonder, what do I do? Do I transfer? Do I not transfer? Mm-hmm. So it's good that there's more research being looked into that, so we kind of have better information about how to proceed if that's the case, if one ends up with mosaic embryos. So thank you. And of course, another big topic in the fertility community and people trying to conceive is always, or perhaps even people planning for the future, is egg freezing. People are talking a lot about fertility preservation when you're younger, to freeze your eggs if you're not sure, you're not even sure you want to have kids in the future or not, but just freeze it. So that way in the future, you have something that you can, consider or use if there's any issues with your own eggs at that time. But then people also say that egg freezing might not be best, that embryo freezing might be better, because then when you thaw the eggs in the future, it might not be as good, or perhaps embryo freezing is much better because when you thaw it, it's usually retains the quality versus egg freezing. So can you share a little bit about that in terms of egg freezing versus embryo freezing?
Absolutely. And yes, egg freezing is great when you're young. If you want to hold off having a family and you're able to do it, I know it can be costly, but it could save you a lot of money down the road if you really do plan to postpone your family until maybe after 35 years of age. Of course, no one can look that far into the future, but if it's something that maybe you have in your life plans, I definitely recommend it if you're able to. It will definitely save you a lot of time and stress down the road, because the younger you are, the more likely the genetic makeup of your eggs is normal. And that's important because we don't want abnormal eggs. They could lead to abnormal embryos. So definitely encourage it. But yes, one thing to keep in mind with eggs is that they're only one cell, where embryos, when you freeze them, are multiple cells, in some cases 150 or so cells. So if one cell in an embryo does not survive the thawing process, that embryo will still be fine. If one cell does not survive the egg thawing process, that entire egg is unfortunately lost. So eggs are definitely more fragile, and we do see lower success when they're thawed for that reason. They're more fragile. I call them more finicky. They're just a little harder to work with. You have to kind of thaw them a little. You have to keep a better eye on them when you thaw them, basically, because if you see any signs of degeneration, you have to catch it really quickly with the eggs, where again, with the embryos, of course, you don't want any degeneration. But again, if you have a couple of cells in an embryo that don't survive the thawing process, your embryo should be perfectly fine. It has all those cells. So definitely something to keep in mind is if you have the option to freeze embryos, say you're with your partner who you plan to be with for forever, for a long time, or someone you really want to have children with, then yes, embryo freezing and thawing will have a better survival rate when it comes to doing the thaw. But egg freezing is still promising. It doesn't mean that all of the eggs will not survive. It just means to go into it realistically, knowing that there's a chance that some of them will not survive. So it's good to have a good number of eggs frozen if possible. So maybe if you have 10 or more, I would say that you're in a good place.
Okay, thank you for sharing that. And it's good to know, because I think when you have a better understanding of, listening to you and saying that, okay, it's one cell versus so many other cells in an embryo, then you can understand why there could be potential issues, with the eggs versus the embryo. So thank you for sharing that. In terms of the IVF process, sometimes there are a few add-ons that people might want to consider, and we're not sure which ones we should opt for. One of the couple of popular ones is assisted hatching. Mm-hmm. So what is it exactly? I think for people who'd like to know, to even have a better understanding, to see if that's something they should even consider or not.
Yes. So assisted hatching is a really actually very simple procedure. What we do is we take a very small laser pulse under a microscope, and we actually make a couple of holes in the shell or the zona that surrounds the embryo. And what that does is it confirms for us that that embryo has a way of hatching if it wasn't already hatching or fully hatched before. So if we thaw an embryo for a transfer and it isn't already thawing or it isn't already hatching on its own, we can perform the assisted hatching, and we don't have to worry about, did that embryo hatch out after the transfer? And the theory behind that is that they say the shell can harden from the freezing and thawing process. So it is important to do the assisted hatching if you're doing a frozen embryo. But one thing to also keep in mind is if you do any genetic testing or PGT, the embryo will already be hatched, because it has to be hatched so we can get in to get the cells inside of the embryo. So if your embryo underwent genetic testing before, it shouldn't need to be assisted hatched before your transfer. So that's one thing to keep in mind. If it's already hatching or fully hatched on its own, there's no reason why they should do any assisted hatching on it. But if you do a frozen transfer, I would say maybe personally to consider doing assisted hatching, only because if the theory that the shell is harder from the freezing and thawing is true, it might be harder for that embryo to hatch after the transfer, and it needs to hatch in order to implant. Other than that, there aren't a lot of new studies about assisted hatching and whether or not they work. I think it's just become something that's so routine in clinics that maybe the studies just weren't worth doing, because, clinics just do it anyway. But, I wouldn't say it's something that's necessary. I know it can be costly and everything, but if it were me, I would do it if I were doing a frozen transfer.
Okay, good to know. Thank you for sharing. And it's good to know that you can actually do genetic testing, and that's already been done. Because, we wouldn't have done. And then speaking about genetic testing, PGT-A and PGT-M, I'm not really sure what they either mean. But can you shed more light a little bit about that, and when should we consider that as an option?
Absolutely. So PGT is pre-implantation genetic testing. So that's the testing that we were talking about earlier. The A stands for aneuploidy, and aneuploidy refers to if the embryos have the right chromosomal makeup. So just briefly, any normal cell in the human body should have 23 pairs of chromosomes, so 46 total, one set from the egg, one set from the sperm. That's what we're all made up of. So each cell in our bodies should have that genetic makeup. But there are situations where the embryos, unfortunately, do not have the right genetic makeup. They have an extra chromosome. They're missing a chromosome. Part of a chromosome is missing. And as we talked about, it's usually 80% or more of the cells in the sample have the wrong amount of DNA. So when we do a biopsy of an embryo for testing, we take about 5 to 10 cells out of that embryo that should have about 150 cells. But the theory is that because the DNA replicates in the embryo, it should all cells in the embryo should have the same genetic makeup. So by only taking 5 or 10 cells, it can represent the entire population of cells in the embryo. And we actually take cells from the trophectoderm, the part that becomes the placenta, and that's because we don't want to damage the part that will become the baby. And the studies are actually pretty promising for that. They do show, we took embryos that were abnormal and we took a couple cells from the inner cell mass and we took a couple of cells from the trophectoderm, and they actually did show concordance, meaning that they did show a very similar genetic makeup. So we feel confident when we do the testing that by taking those 5 to 10 cells, we're taking we're getting a good representation of the entire cell population of the embryo. That's just a little side note. So what the genetic testing does is it actually looks at what percentage of the cells in that sample have the right amount of DNA. So that's where we get those percentages from. If 80% or more cells in the sample have the wrong amount of DNA, that embryo is considered aneuploid or abnormal. And that's where that PGT-A comes in. PGT-M is a little bit different. The M stands for monogenic, and that is a certain type of genetic disease. An example might be cystic fibrosis, sickle cell anemia, things like that, but that's not very common. So we only do that in situations where either the sperm or the egg source is a carrier of a genetic condition or affected by a genetic condition. But normally what happens is people, before they do IVF, they'll do carrier screening. It's usually a blood test or a saliva test, and it can determine if you're a carrier of a panel of genetic diseases, again, including cystic fibrosis, Huntington's. There's a whole slew of things that they can test for. If they find that the egg source and the sperm source both are carriers of the same condition, then I believe 50%, there's a 50% chance that the embryo will be affected by that disease. And in some cases that can be lethal. I mean, that could mean that the child could have cystic fibrosis or some other kind of genetic disease. So the carrier screening is important for that reason, because it can help determine the percentage or chance that the offspring will be affected by that particular disease. And that's the only time that we really do PGT-M or in those situations. So it's not routinely performed, but it can also be performed with PGT-A. So for example, you might have an embryo that's not affected by cystic fibrosis, even though you carry both sources carry it, but it could be genetically abnormal, in which case, although it's unaffected, it's still genetically abnormal.
Good to know. Thank you so much. That was really helpful. And, when choosing a clinic, what are some of the areas that we should focus on with the embryologist? Because, we all talk about choosing a fertility clinic. What should you do? Check if they're responding to your emails, see how the environment of the clinic is, see if the doctor seems to understand and listens to you. But what about the embryologist? Are there any questions we should be asking about that, or any attention we should be paying to what happens in the lab? Should we be asking any questions about that at all?
You can definitely ask some questions about the lab. The things that I would maybe ask are, how many cycles do you do in a year, and how many embryologists do you have on staff? And that every embryologist is very different. We all work very differently. Some of us work better at a faster pace, like we work very efficiently at a fast pace. Some embryologists prefer to take their time and really focus on each task, just like people. We're all different. Yeah. And so, if you have an embryologist who really takes their time, but there are a lot of procedures, then maybe that embryologist is overwhelmed or something. Vice versa, if you have an embryologist who works quickly, but they don't do a lot of cases, then, that embryologist will get everything done. But there could also be multiple embryologists working. So a lot of labs that I work with have, 3, 4, 5 embryologists working at a time. So that's usually not a huge issue, but it is something that you could ask, how many embryologists do you have working versus how many cycles are you doing at a time? And I think just a really important thing too is just to ask the overall success rates for the clinic. And a lot of that boils down to the lab too. Like, what is your ICSI rate? What is your blastocyst rate, meaning how many fertilized eggs develop into blastocysts that can be transferred. What is your pregnancy or transfer rate, how many embryos implant, and that sort of all boils down to the lab because they're the ones who are doing a lot of those procedures. So that's certainly something that you could ask too. That even might be more important than, how many people are on staff versus how many cycles.
Wow. Yeah, thank you. Thank you for sharing. These are really important questions that we might not even consider at all because we're so focused on other aspects of it. So thank you. That was really helpful. And when you talked about success, your opinion as an embryologist, any insights on how we can generally increase chances of success when going through the IVF process?
That's a question I get asked a lot. And
Sorry.
Sorry. Yeah, so I get asked that question a lot. And for sperm, there are ways to improve quality. With eggs, it's a little bit more difficult because women are born with all of the eggs that they'll ever have. And so women, their eggs are sort of at their prime in their early 20s to mid 20s. And that's evolutionary, meaning that not so long ago, it was more common for women to have children younger. Now, as we're advancing as a society or as a culture, our bodies just haven't kept up with that yet. Our eggs are only meant to be there, or our eggs are only meant to be at their most optimal state, in our early to mid 20s. And then from there, the quality actually starts to slowly decline. And then we see that rapid decline normally around 36 years of age. And then, of course, women, the average age for menopause is, I think, about 50 to 51. So when you think about it, it's a good amount of time, but it's not, it's not huge considering that women are waiting to have their children or start their families or whatever it may be. But it's hard to improve the quality if it's already been declining, is what I'm trying to say. So we have this natural decline and there's really no scientific way to improve the quality after that, but there might be ways to maintain the quality. So it might be good to start when you're younger with good lifestyle changes like a healthy diet, a normal exercise routine. It's, all the things they say in life, like don't smoke, moderate your drinking, try to walk, eat better, just everything that you can do for a healthier life. Avoid stress, try to avoid toxins. For the eggs, it could work. It's just not scientifically proven that it can actually improve the quality, but a lot of people have found success with this. So that's wonderful. For men, it's a little bit different. Men actually produce sperm from puberty on. And so they go through constant production of sperm. So you can actually improve sperm quality because if you start these changes early, for example, a lot of men might need to take a certain supplement or a hormonal therapy regimen, right? Because maybe that's the problem they're having is a hormonal imbalance. Now, if you can go in and you can fix that early, about 90 days later, which is about how long it takes for new sperm to be produced, you can actually see changes in their quality or their motility. So those are good ways, I think, that you can help to at least maintain, or at least in the male case, improve the quality, which can then improve your success rates. But again, not scientifically proven for women yet, though I am hopeful. But I think the most important thing is being able to maintain the quality as early as you can. So making those good, healthy choices early could help you down the road because it can help to at least maintain that good egg quality instead of it declining at a faster rate.
Yes, thank you. Oh, it's so, don't we wish we were just like men that had the chance of continue to produce eggs every 90 days as opposed to
yes. I completely agree. It's not fair.
Yeah, no, they have to do all the extra work for the IVF as well. But anyways, it's good that there's more research going into it. And again, like you said, if there's ways we can maintain the quality, at least keep eating right, and we can't do anything about the age, that's going to happen anyways. So we can focus more on the things that we can do and try to maintain a healthy lifestyle as much as possible.
Right.
So, it's been really amazing listening to you, but I just wonder if there's anything else that you'd like to share with anyone considering IVF right now, anyone listening and considering IVF, any other information that you'd like to share as an embryologist that you think might be helpful?
I would say a couple of things. If you're, beginning the process, do, make sure that you feel comfortable at your clinic. Don't be afraid to ask for the success rates for your particular situation. So an IVF clinic might have a good success rate, but they might also primarily see women in their mid-20s who have perfect eggs or perfect sperm. So it's good to ask specifically for your success rates. Like, what is my rate for a 35-year-old with PCOS and my husband has low sperm count? Like, what are my chances of success there? Those are going to differ from someone who's maybe 25 with, perfect eggs and perfect sperm. They're going to be different rates. So that's something to keep in mind. And it's really important that you feel comfortable. You will spend a good amount of time at your IVF clinic, and especially when you get into the IVF procedure, you might be there every other day. So it's important that you feel comfortable. It's important that you feel heard, that your questions are answered. Confidence is key. And I would also just say to always set realistic expectations. I think a lot of people think that because they're going to do IVF, they're guaranteed a successful outcome. And that is often, a lot of people, for a lot of people, that is completely true. IVF is what they needed. They have a successful outcome, but just know that your first cycle might not be successful. Your second cycle might be, or maybe you require a couple more. And in some cases, unfortunately, you might have to turn to alternative options, because going into the IVF with your sperm or your partner's sperm or your eggs or whatever it may be, that just might not end up being successful. You might have to consider looking into just alternative options at that point.
Yeah, thank you so much. That was really, really helpful. And that's so important that you say that, because again, oftentimes with IVF, you hear that it's always, it's almost like it's a guaranteed outcome, but it's not reality. So it's good to go in that with realistic expectations and just be willing to know what the potential options might be or available things to consider. So thank you so much.
Of course.
It has been amazing having you on here, Jessica. I feel like I just went to school, and I'm sure everyone that's listening as well feels the same. We have learned so much, because again, the embryology part of the whole process, we just don't really understand. I mean, people have so many questions, you don't know who to ask. So to have you come here and join us and answer many questions has been amazing. So thank you so much.
Of course.
For your time today, for taking the time to answer all our questions, and for also creating your page, because again, your page, if there's anyone listening and you're not following at Explaining IVF, you need to follow Jessica's page, because it really is amazing, so informative. I love your videos. I love how you make everything seem so simple and understandable for anyone reading it. So thank you so much for taking the time to do that, because you don't have to, you already do your work at work, but now you're doing this just to educate people and create awareness. So that's amazing. So thank you so much.
Thank you. Yes, I love doing it. So
yes, thank you. Yeah, so you're such a blessing to the community. So thank you for all that you keep doing. And all your details will also be in the show notes for people to follow you and reach out to you. What's the best way to reach out to you? Is it on Instagram, or how can people connect with you if they want to reach out to you?
Yes, I've shifted most of my attention to my Instagram, just because that seems to be where I connect with the most people. So feel free to send me a message about any questions you might have. I'll do my best to answer them, but I can't answer everything. A lot of stuff does have to go through your doctor, but I'm happy to at least try to answer or point you in the direction that you would need to go to get the answers you need.
Thank you very much, Jessica. And thank you again for being here today. And we hope that we can have you again in the near future to further help us with more questions. But we thank you so much for all that you're doing to help and support the Trying to Conceive community. You're such a blessing. Thank you.
Thank you.
Thanks for joining us this week on the Fertility Conversations podcast. If you enjoyed this podcast, please give us a 5-star rating and subscribe. Follow us on Instagram at Fertility Conversations. If there are any topics you would like to have discussed, please send an email to fertilityconversations@gmail.com. Be sure to tune in next week for our next episode. Thank you again for listening. Take care of yourself and do stay hopeful.